Cancer is a major problem. It afflicts about 1/3 of the population, and unfortunately 1 in 4 individuals die from cancer. In spite of advances in the treatment of cancer, the mortality from cancer in many cases has remained static. We believe that there is need for a new approach to deal with this problem, and we believe that tumor immunology is a particular and exciting new approach.
I met Professor Ehrman some 10 years or so ago. He had a very interesting project. The idea was to use fragments of proteins that normally expressed at higher levels only in tumor cells in order to vaccinate patients and therefore induce an immunological response for rejection of the tumor in the same way the immune system normally gets engaged and reduces a bacterial or a viral infection.
September 2000, when I was diagnosed with breast cancer, I met Professor Ehrman, who became my surgeon and consultant. During the year's treatments for breast cancer, which included surgery, chemotherapy and radiotherapy, I discovered, along with Dennis Firth, my co-founder, that he was in fact leading a research project into immunotherapy. We learnt more about that as we met Professor Ehrman and decided that perhaps it would be the thing to take on.
We're now in the process of looking at a new and novel approach, vaccinating patients who have advanced cancer with a range of small molecules called peptides. We use these peptides to inoculate the patients, generating anti-cancer immune defenses, which once generated in the body will travel to the cancer, lead to its destruction. There have been a number of studies published in the literature, trials in patients with advanced cancer in melanoma and prostate, where immunotherapy has been shown to improve patient outcomes.
In order to generate the vaccine, we have to take fragments of these proteins that are overexpressed or uniquely expressed in tumor cells, and these fragments are actually synthesized in a laboratory. As it happens, these particular fragments are synthesized in a laboratory in the States, and the fragments are sent to us as a dry powder. They need to be dissolved in solutions suitable for injection. This is done inside an isolator. The individual peptide solutions need to be mixed together. They need to be controlled and regulated for pH and other factors, and eventually generate the solution that actually goes into the vial that we send to Professor Ehrman for injection into patients that qualify for inclusion into these early stage trials.
The principles underlying this approach are not dissimilar to that of you as a patient or as an individual going to your GP and being vaccinated against flu, so that you generate particular antibodies or specific cells which are then able to kill the viruses or bacteria should they occur in your body. And hopefully in the long-term future we may be able to perhaps vaccinate patients and prevent the disease from occurring.
And we know that with the new type of treatments there will be little or no reaction, which in itself has to be amazing for the patient themselves and their families.
We hope to enroll 30 patients between the ages of 18 and 85, males and females, with a whole range of different solid cancers. They will be referred by their treating surgeon or oncologist based at the United Lincolnshire Hospitals NHS Trust or Nottingham University Hospitals NHS Trust. We will be inoculating them with these small molecules on a 3-weekly basis for 8 courses. And on the day of the vaccination, the patients will come in and have the vaccine injected. This will be given intradermally into the skin around the periumbilical area, the belt line. One of the important elements of this study that makes it rather unique is that we will be inoculating the patients with 7 peptides, which will mean that we can treat any patient with any type of cancer. The study will probably take between 2 to 3 years to complete. This is a complicated study which is quite expensive. We have to have the peptides prepared in a very special environment, laboratory. This has been done in the laboratory overseas. These then have to undergo necessary further rigorous testing in the Raine Institute in London. We have to use various medications and drugs. All of this adds to the costs. We have to pay for patients to travel to have the inoculations carried out. We have to pay for laboratory investigations and for people working on the trial.
Donations are of course crucially important to the development of this work, particularly so now where, ironically, even though the final product, if successful, is going to be substantially cheaper and more easily available to large populations of patients, these very early stages are extremely expensive because the synthetic peptides are very expensive and because the facility and equipment that needs to be used in order to generate these very small doses of vaccine as opposed to mass production makes the process very expensive.
To date, we have had funding predominantly from an organisation called CANDLES, which has been specifically set up in Lincolnshire to support my research and those of my colleagues so that we can do these sort of studies, particularly in patients that are living in Lincolnshire.
For us, not only is it a challenge financially, a major challenge, it's an exciting time to see all his work of over 25 years and our 12 years come to some fruition.
Although it is entirely possible and likely that later stages of the development of this vaccine would receive support and donations, maybe from other organizations, government bodies, possibly the pharmaceutical industry, these early stages are crucially dependent on charitable support.
The 2-year target is 300,000 in 2 years. It's a big ask. It's an awfully big ask for the people of Lincolnshire, and so far they've given us no reason to think we can't do it.
In the last 10 years there's been a great increase in interest, both clinically and scientifically, in vaccination, and CANDLES have enabled us to go through this very difficult period and produce information and data which is crucial to enable us to do this particular study in the UK.
I would like to think my grandchildren will perhaps never really worry about who amongst us will be diagnosed with cancer.